In this video we will discuss about the CD4 plus T cells regulation. So how these CD4 plus T cells are regulated within the body. So we will first discuss its upregulation mechanism, and then we will discuss its negative regulators also. So the activations or the upregulations of this CD4 plus T cells starts with this IL-7 when it interacts with these T cells. So when this IL-7 interacts with the T cells, it will lead to the phosphorylations of the intracellular STAT5, and it will cause this phosphorylated STAT5 formations in the cytosols of the cells or T cells. And this phosphorylated STAT5 state of this STAT5 will help them to translocate into the nucleus, where it can interact with the promoters in a way that it will downregulate the apoptosis of the T cells. At the same time, it will upregulate the proliferations of the CD4 plus T cells. This is how CD4 plus T cells are positively regulated with this interleukin 7. However, this PLA G1B, it is also secreted inside the human beings naturally, and the upregulations of this PLA2 G1B will cause release of this arachidonic acid or A release, and this arachidonic acid will then be interact with the membrane of the CD4 plus T cells to form abnormal this abnormal membrane microdomain or aMMDs, and this aMMDs has the capacity to prevent the IL-7 interactions to this IL-7 receptor. And once this IL-7 interactions to this IL-7 receptors is prevented, the intracellular signaling pathways that leads to the CD4 plus expressions will also prevented. That is shown here, that phosphorylations of this p-STAT5 will not occur, and thereby the promoters, it will not change the promoter functions, and it will affect not only the proliferations capacity of this CD4 plus T cells, but it will also affect the survival ability of this CD4 plus T cells, as now the apoptosis related factors or proteins will be upregulated because they are not been controlled by p-STAT5. So this is in general how the CD4 plus T cells are being regulated by this interleukin 7, and this abnormal membrane microdomain, it it can interact with this IL-7 receptors, as we have discussed. So these things are usually formed in the pancreas. So whereas it can act as a negative regulators for the CD4 plus T cells formations. On the other hand, when this PLA2 G1B is not present, then it will cause the IL-7 interactions with this with its receptors. So in the case of this autoimmune disease, one of the conditions that might occur if this PLA G1B is not been secreted normally, in that case there is a continuous activations of the T cells with the presence of IL-7, and it will cause the conditions where the immune system might attack their host normal cells also, or it will cause the inflammations that will result in the abnormal side reactions, abnormal or side reactions. So this is one of the conditions where autoimmune disease can be formed with the problem in this PLA G1-2B. On the other hand, if this IL-7 is not receptors are not working or they are in the mutated form, then it will prevent the activations of the T cells, and this will leads to even more problems, that the immune systems of the patient is not healthy and it might get infected with different kind of bacteria or virus that are presented in the environment, in its nearby environment. So these two conditions can be easily understood from the CD4 plus T cells mechanism. So if you need to understand more about this CD4 plus T cells regulations or its mechanism, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.